It is my pleasure to be here. I've been coming many years off and on to the LA LACNet's conference. So we are going to jump right into some complicated stuff on clinical trials. But think of it sort of as a clinical trials 101. We're really going to try to dissect how to think about a clinical trial. See if I can get this working correctly. All right, so we're going to talk some clinical trial basics, definitions, how we define success in clinical trials. We'll talk about some ongoing and future NET trials that I think really what I want to leave you with is a lot of optimism and hope that we have a lot of future advances coming down the road, and then how you can learn about trials. So this is to just set the stage and again, really give you a take home of how much progress we've made in the last few years. So in the 80s, we had streptozocin and octreotide, and then it was a bit of a ghost town for 20 to 30 years before we had FDA approvals of other agents. And since 2011, you can see this explosion on the right side of your screen of all of these new FDA approvals. And so it's been a really exciting time. We have lots of, I always say to my patients, we have more tools in our toolbox, and that's a really good thing to have, and that will certainly continue. All of these drugs, though, had to have gone through clinical trials in order to get FDA approved. So we'll talk about what those are. So this is the definition from clinicaltrials.gov. So clinical trials are research studies that explore whether a medical strategy, treatment, or device is safe and effective for humans. Why are they important? Well, trials are a key research tool for advancing medical knowledge and patient care. And clinical research is done to learn whether a new approach is safe and effective, and which treatments or strategies work best for certain illnesses or groups of people. And so trials are really done often for a specific disease, so for neuroendocrine tumors specifically. Now, what's interesting is only about 3% of US adults with cancer participate in clinical trials. This is quite different than in the pediatric population, where well over 80 to 90% of children with cancer participate in clinical trials. So for those of you out there who have been a clinical trial participant, I thank you. This is really a way that will move the field forward. So how are trials different than standard of care that you may be able to get through your local oncologist? Well, standard treatments, the side effects generally are well known. The drug dose and intervals, the frequency of your doctor's visits, how often you get labs, that's often very flexible, and you usually can get that treatment with your local or community oncologist. Clinical trials, the side effects may be less well understood. The drug dose intervals, doctor visits, labs are usually at fixed intervals, so there's much less flexibility in terms of, hey doc, I'd like to go take a vacation. Can I come back in 3 weeks and get that treatment? The answer on a trial is usually no. Treatments usually are required at the institution that runs that trial, and more people are involved. So there is a research coordinator and often a data entry person in addition to your regular clinical team. I actually see that as a positive with more people thinking about you. So there are a number of different ways clinical trials can happen. So a sponsor of a trial really refers to who thought of the clinical trial idea. It does have somewhat to do with who funds the trial, but there are 3 key sponsors. So there are investigator-initiated trials. This is when an individual doctor may think up an idea and then try to get funding from a pharmaceutical company or another or a nonprofit. These are often much smaller studies. We'll talk about what the phases mean in a moment, but phase 1 and 2 are smaller. They're usually available at a single academic institution. Idea is initiated by that academic physician, and the funding and/or drug supply is often from industry. Industry-initiated can be any phase of trial, usually involves many sites and is sometimes international. This is often a way to get big studies done, often in rare diseases. And the idea is initiated by industry, and funding is from the pharmaceutical company. And then cooperative group trials are federally funded. These can also be any phase sponsored by the NIH or the National Cancer Institute, also open at many centers, and the funding is federal. So let's talk about the phases of trials. So before we even get into humans, there often have to be preclinical trials or laboratory-based studies that are done to determine, does the drug work in mice or another animal? In phase 1, we're just looking at, is the drug safe? Often it's any type of tumor is eligible, usually a small number of patients, and the side effects are defined and the best dose is determined. So the purpose of this type of trial is not to look at efficacy, it's just safety. If a drug passes phase 1, it's deemed to be safe and you've picked the right dose, you move to phase 2. And you have a preliminary test of efficacy also in a small group of patients. It's usually limited to a specific tumor type. And then phase 3 tests efficacy, often in a randomized way, meaning one group of patients gets the experimental drug and another group of patients gets either a standard drug or a placebo. I get lots of questions about, but doc, I don't want to be on the placebo. Well, it's described what that risk would be to you if you were to consider a placebo trial. And placebos are only ethical if doing nothing is considered standard of care. So lots goes into, and it's really explained quite well in the informed consent process for a trial. Phase 4, after FDA approval, we collect additional data on safety. So every drug has to go through phase 1, 2, and 3 before it gets to FDA approval. This process can be quite arduous. It takes about 10 years to get from beginning to end of that process. And even if a drug is FDA approved, it does not guarantee benefit for everyone. So when I talk about standard treatments and clinical trials, I think it's really important to acknowledge that though we may have data based on a clinical trial for even standard of care drugs, or even if you think that immunotherapy trial sounds so exciting, we really just can't guarantee benefit. We can talk about risks and benefits, but it's not guaranteed. So even within a trial, there are specific types of designs. So phase 1 trials usually test increasing doses. So you might start with a small dose. The next group of patients, you maybe test a slightly higher dose. A single arm trial means that everybody on that trial gets exactly the same treatment. That's usually a phase 2 trial. And then a randomized trial, as I said, could be one or more arms where each arm gets a different treatment. And the randomized trial is really the gold standard. That's really what's required to prove that something is better than a standard of care. So for NETs, what matters when developing a clinical trial? So there are things called eligibility criteria. I talked about that briefly as it related to the NET Vitals checklist. So which NET? Is it pancreatic NET or small bowel? Which grade? Is it growing or not? Are there certain prior treatments that would eliminate you from being eligible for that trial? And then how do we define success on a trial? So progression-free survival, and we're going to actually dive deeper into each of these, but how long does it take for the tumor to grow? Overall survival, how long do patients live? Response rate, how much does the tumor shrink? And it's also becoming increasingly important to include quality of life measures in clinical trials, often through patient-reported outcomes, and also safety. So we are going to take the NETTER-1 trial and dissect it a little bit to learn a little bit about how these measures of success are defined. So the NETTER-1 trial took 229 patients with mid-gut neuroendocrine tumors. Those are mostly small bowel neuroendocrine tumors. Half got the radio-labeled octreotide, that's the lutetium dotatate, and half got high-dose octreotide at 60 milligrams. So you might think, I have no idea how to look at that graph on the right, so let's look at this a little bit more closely. So the horizontal axis in these figures is usually time. In this case, it's months. And the vertical axis refers to progression-free survival. So it is a percentage of patients that do not have progression, so they are free of progression. And that dotted line refers to the median, so kind of the halfway mark. And that's often, median progression-free survival is often a primary outcome for NET trials. So separation of the curves is good. So if that's kind of a key take home for how we look at these curves. You want to see that there's a difference. Each of those lines represents one of the arms of the trial itself. And then when we look at sort of how they're measured, we want to see that that experimental arm does better. The progression-free survival is longer compared to octreotide. So that's true in this case. And interesting, let's see if I can get this to work up here. So for octreotide, the median progression-free survival was 8 months. So you can see where it hits that line here. The ludotatide arm had not actually hit the median. It's estimated to be about 3 years or 30-ish months. And the hazard ratio refers to how much ludotatide reduces the hazard or reduces the risk of progression. And it reduces that risk by about 80%. So this is a very positive trial. We're fortunate to have Dr. Strasberg in the room, who is the lead on this study, and I'm sure he'll speak to this a little bit later. Okay, so understanding tumor measurements. I get asked this a lot. This refers to like how do the tumors shrink or change over time. On a clinical trial, we use criteria called RECIST criteria, Response Evaluation Criteria in Solid Tumors. It's an acronym. And the key features of these criteria is that the goal is to measure representative, but not all tumor lesions over time. We typically measure a maximum of 5 and a max of 2 per organ, and we'll walk through this in a bit. We measure the longest dimension of that tumor spot, and then we look at that and how it changes over time. And it's probably easier if we just move to the next slide. I have a little cartoon to kind of go over this. So imagine that this is a cartoon of a liver. The yellow spots are 2 tumor spots in the liver. We've measured the longest axis of those 2 spots. And at baseline, the sum of the longest diameter is 9 centimeters. So then after 3 months of treatment, those 2 spots shrink. We measure those as well here. And the percent change, it has shrunk 17% compared to baseline. That officially is considered stable. Anything that is between 20% growth and 30% shrinkage is considered stable. And then the patient gets another 3 months of treatment, and the tumors have shrunk further. And we measure that here in this, this last row column. And the percent change from baseline here is 44% shrinkage compared to baseline. That's considered a partial response. And on the NETTER-1 trial, just to kind of pull it all together, they then summarize all of those best responses. So a partial response or a complete response is considered an objective response for purposes of a trial. And there was an 18% response rate. So that meant that on NETTER-1, about 1 in 5 patients had a respectable amount of tumor shrinkage on this study, and compared to only about 3% on the octreotide alone. Just to move on and talk a little bit about clinical trial myths. These are things I get asked a lot about in clinic. So I get patients will come in and say, well, but clinical trials are better than standard treatment. I know I'm on a drug that's working, but I want that new thing. And I think this happened a lot for NETTER-1. And just as a reminder, the grass is not always greener. I think that if you are on a therapy that is working, I generally advise to not jump ship from that until we really need it. Another myth is that clinical trials are only for patients who have no other options. That's also not true. We have trials for almost, many, many phases in your disease course. Another is that I might get a sugar pill or placebo instead of a real drug. Well, you will never be duped into that. That is always completely reviewed prior to enrolling on a clinical trial in terms of what your chances would be of perhaps getting a placebo. And trials are not for everybody. If you are not comfortable with the idea of not being able to choose which arm you're on a randomized trial, then it may not be for you. Also, I'll have a lot of extra costs that my insurance won't cover. There are, all clinical trials have a different portion of what aspects of that are covered either by the trial itself or through your regular standard of care and insurance. So there may be co-pays that are associated with a regular doctor visit. Those may still be hold true if you're on a clinical trial. And then once I decide to participate, I won't be able to change my mind. Also not true. You can do and come off of a trial at any time. Really, the care of the patient is what matters most, not the care of the trial. All right, what happens when I go off of a trial? So patients go off for many reasons: progression, side effects, personal preference. Your doctor will discuss other treatment options, including both standard options and trials. The frequency of monitoring may be less on a trial, and also those tumor measurements are not considered standard outside of a clinical trial. And I just want to acknowledge that, because I have had a number of patients say to me, "Doc, where are my tumor measurements?" if we end up moving them from a trial onto standard of care. I think that is something that I'm a big advocate of, doing quantitative measurements even off of a trial, but I think that there are some of these key differences between standard of care and a trial. So categories of drugs in NET trials are many. There are somatostatin analogs, biologics. There's a new trial that will start soon with one called cabozantinib. There's chemotherapy, PRRT, and liver-directed therapies. I'm going to spend just a couple of the last minutes going through a few examples of some of the trials that we have available. So this is a table that goes through some of the ongoing studies. They all have a funny name here called the NCT number. Every trial has to be registered on the clinicaltrials.gov website. And I'll put in a little bit of a plug and can give you a bit of a teaser. So this first trial, a lot of people are familiar with CAPTEM or TEMCAPE as being used for pancreatic NETs. We've just completed a large pancreatic NET clinical trial through the cooperative group system that was federally funded. I'll be presenting that at ASCO this year, which is a big oncology professional meeting. This was just made public this week, so I can share this. The TEMCAPE, it was compared to Temodar alone, and the TEMCAPE arm did considerably better. The median progression-free survival was about 2 years on that, which is really exciting. So I think that will be an important, another tool to add to our toolkit for patients with pancreatic NETs. And I'm not going to go through all of the details. This is really intended to show you that there are so many committed NET researchers in many different arenas, and I anticipate that many of these will also become practice changing. And so lots of clinical trials. So when you see your treating physician, it's good to say, "Can you tell me about clinical trials?" And if they do not know about clinical trials, then you ask to be referred to either an academic center that may have more trials, and I think that's a perfectly reasonable question to ask. I'm going to jump to this. I think this is a kind of a practical piece. So how can you learn about clinical trials? I would say the main one is through clinicaltrials.gov. That website is a little bit cumbersome. We'll talk just briefly about that. NetRF has trials listed on their website. Many of our own institutions have their trials listed on their website, so there are a lot of ways to find out, but it's really your treating physician that can help guide you. You're not expected to try to find trials on your own. So let's just walk through this for a moment. So this is the clinicaltrials.gov website. If you were to write in neuroendocrine tumors, you would come up with like 2,500 studies. So how do you sift through that? There are ways and filters that you can say, I just want to look at studies that are actually open for adults, phase 1 or 2, so you can filter that some. As I'd mentioned before, this number here, the NCT number, is a unique identifier for all trials. So that gives you, if you hone in on that, it gives you more information about the trial. There are a lot of different ways to view this website. I find that the tabular view is really the easiest to read, and it gives you some additional details about the trial itself. So in terms of looking to the future, what does the future landscape look like for clinical trials? I think that many of us are interested in looking at how to improve and optimize PRRT. So how can we improve on 177 ludotatide? I know there are going to be many conversations later today with some of our experts in the room. There are novel ways of administering PRRT and perhaps even new indications like paraganglioma and pheochromocytoma and lung mets and perioperatively. So I think very exciting from the physician community. We are excited about this treatment and want to figure out how to best use it. Immunotherapy, I got asked lots of questions about this. I think it's still not ready for prime time in NETs. We do have some ongoing clinical trials and some new ones. A new one specifically, there's a new bispecific antibody trial that essentially helps marry and bring together a NET cell and a T cell, which is part of your immune system. And then personalized therapies. How can we learn about your individual cancer through genomic profiling to really help tailor treatments to you? So I think this is some of what the future looks like. So take-home points, many advances in the last 7 years. It's been a really exciting time to be a researcher in the field and very rewarding, and really a time where I can say to patients, you know, you have lots of options. And clinical trials are critical for progress in NET treatment. We can and need to do studies in rare diseases. And then I'd mentioned all of these therapies that are likely to be incorporated. And again, thanks to those of you who have already participated in trials. I'll end there.